It gives me great pleasure to introduce our speaker. He is Chief of the Division of Rheumatology in Northwell Heath, New York. He's Professor of Medicine at the Donald and Barbara Zucker School of Medicine at Hofstra, Northwell, New York. He's regarded as one of the senior rheumatologists in the New York metropolitan area. For 20 years, he served on many committees of the American College of Rheumatology, ACR, and was named as Master of the College in 2018. Dr. Fury's research focus has been in systemic lupus, SLE, and related diseases such as the antiphospholipid syndrome. Specifically, he has played an instrumental role in drug development and clinical trial design in SLE and lupus nephritis. And now he is giving us his Sunday morning to talk about lupus clinical trials and why so many fail. He's here from New York. Professor Fury, the floor is yours.
Thank you. Let me share. Okay, I think we are set. Yes, my name is Rich Fury, and I'm the Chief of Rheumatology at Northwell Health. And for those of you who don't know the New York area, Northwell Health is a conglomerate of about 24 hospitals covering Long Island, New York City, Westchester, Queens, Staten Island, Brooklyn. I think we have the whole area covered. And for rheumatology, we're rather big. We have about 45 full-time rheumatologists throughout our network. My passion is to take care of lupus patients, and I've been doing that for close to 40 years. And then about 25 years ago, we started doing clinical research trials in lupus. And I must say that has been incredibly challenging, but we're finally starting to see the rewards of our efforts. So though I think in the title is something, why is lupus research so complicated? Well, it is complicated, but I think we're finally cracking the nut. So what I thought I'd do is talk about developments from the American College of Rheumatology meeting. And these are pretty fresh findings. Our meeting was just about 2 weeks ago. And then at the end, talk about some of the challenges that remain for lupus clinical research. So I don't have to tell this crowd, there is no disease more heterogeneous than lupus. And this is a collage with some of my patients and some that I've gotten from the American College of Rheumatology website. But you see all different clinical manifestations. Neonatal lupus in the top left, Raynaud's, that PET scan on the top row towards the right is a patient of mine with chorea and antiphospholipid antibodies. You see vasculitis, many different kinds of rashes ranging from acute cutaneous lupus to discoid lupus. We have immunofluorescence of the glomerulus in the second row on the right. Vasculitis resulting in gangrene, arthritis down on the third row, heart disease, valvular disease in the bottom row, and again, many different kinds of rashes. And there's pathology intertwined as well. And I could go on and on with many more pictures, but challenge me, there is no disease more heterogeneous than lupus. And that's led to a lot of challenges. So it's difficult to prognosticate because no two patients are alike. And you'll notice that in the top of the slide, I have snowflakes. And the reason for snowflakes, the Lupus Research Institute many years ago asked me to critique a brochure that they were developing. And it was, like fingerprints, no two lupus patients are alike. And I said, well, it gets the point across, but it's a little too criminal. Let's change it to snowflakes. Anyway, that's why there's snowflakes up there. So it's difficult to prognosticate, though I think we have a good sense of who has the potential, what kind of patient has the potential to become very sick. based on demographics, based upon their autoantibodies. And not that we have a lot of different medicines available to us right now, but the question is, which medicine for which manifestation? So for someone who has arthritis, which immunosuppressive might we pick? For someone who has skin rash, would you pick that same drug or not? And it's made the development of therapeutics very challenging. I mean, how do you come up with a drug that can treat all of lupus? And then on top of that, lupus nephritis, which has a very different treatment paradigm. So it's made clinical trial design very difficult. All right, but we've had some highlights. And I'll just review 3 of what I consider the major highlights over the last couple of decades. And one is the introduction of mycophenolate mofetil. The history of mycophenolate mofetil in lupus goes back to Hong Kong, to a study done by Dan Chan, where he enrolled 64 patients with proliferative lupus nephritis and compared mycophenolate to cyclophosphamide. Cyclophosphamide was basically the standard of care for lupus nephritis, for proliferative nephritis. Whether you like the NIH protocol or the Euro Lupus protocol, cyclophosphamide was the way we treated patients with kidney disease. But we don't like cyclophosphamide. In fact, a lot of the drugs, in fact, all the drugs that we use, we have a love-hate relationship with, including cyclophosphamide. The comparison was between oral mycophenolate, which we thought would be much safer than cyclophosphamide and might provide similar results. And in fact, that's what he showed in this 64 patient study. And you can see on the right-hand side of the slide, the complete response rates in the 2 groups. They're identical. They're very high, but they're identical. And that prompted Ellen Ginsler from Downstate in Brooklyn to do an FDA-sponsored study where she not only included patients with proliferative lupus nephritis, but also included patients with membranous nephropathy. And there you can see that there was numerical superiority of mycophenolate over cyclophosphamide. And then finally, that led to a worldwide study called the ALMS study, ALMS, Aspreva Lupus Management Study. And that was authored by Jerry Appel, a nephrologist from Columbia in New York. 370 patients with proliferative nephritis, but also pure membranous was allowed. And you see the complete response rates between mycophenolate and cyclophosphamide were the same. Now, what's rather striking is that the difference in response rates in the 3 studies, but what was common to all 3 studies was that mycophenolate performed just as well as cyclophosphamide. The hope was that the FDA would approve mycophenolate for the treatment of lupus nephritis, but through idiosyncrasies of FDA rules, it could not be approved. Though it's widely used now, and with the generic available, the cost has come down. The ALM study had a maintenance phase. After 6 months, patients were re-randomized, and the comparison was between mycophenolate and azathioprine. And what was the primary endpoint was a composite, basically of worsening or of failure. And you can see half as many patients treated with mycophenolate failed compared to azathioprine. So this was clearly a highlight. I mean, I rarely use cyclophosphamide these days. My go-to drug is mycophenolate. And in fact, we're seeing a lot of mycophenolate used for extrarenal manifestations. Another highlight was the approval of belimumab. And I remember that vividly because the FDA Advisory Committee was on my birthday in 2010. So it's been about 10 years since the FDA Advisory Committee meeting and almost 10 years. It was March of 2011 when the FDA approved belimumab. And that was based upon these 2 studies, BLISS 52 and BLISS 76. BLISS 52 was the extra outside the US study, and BLISS 76 was the US Western Europe study. But they were both successful studies. And these two pivotal studies led to the approval, not only by the FDA, but of other regulatory agencies around the world. And this was history making, because this was the first time a drug succeeded in lupus via the route of a randomized controlled trial. Now, there were FDA approved drugs, but they were all grandfathered in. Hydroxychloroquine was grandfathered in, steroids were grandfathered in back to 1952, and anti-inflammatories, or I should say aspirin, was grandfathered in. So this really made history. And then I list hydroxychloroquine. We used to use hydroxychloroquine essentially for our patients with rash or arthritis, but there were a lot of data accumulating that every lupus patient should be on hydroxychloroquine because it had benefit in multiple different domains, and I've listed those here. It increased survival, it reduced lipid levels, it has antithrombotic effects, it reduced cumulative damage, it prevented flares. So the rule is everybody should be on hydroxychloroquine. Is everybody on hydroxychloroquine? No. I mean, I've been caught with my own data. About 2/3 of my patients are prescribed hydroxychloroquine and no more than that. I can tell you why. Some who are really sick say, well, why do I have to take, in addition to high doses of steroids and an immunosuppressive and maybe a biologic, why do we need yet a fourth drug? And then the patients at the other end of the spectrum who are doing just fine, almost in remission, say, well, why do I have to take a medicine? I am doing well. So that probably accounts for most of my 1/3 of the patients who are not on hydroxychloroquine. Then a more important issue comes up. Those who you prescribe hydroxychloroquine to, are they taking their medicines? And Natalie Costa-Doet from Paris has shown us data for many years that there's a large percentage of patients who were prescribed hydroxychloroquine, but when tested for their blood levels, the blood levels come back 0. So non-adherence or non-compliance is a problem, not just for hydroxychloroquine, but for many of our drugs. Well, the big question is, what will the next 10 years bring? So what I'd like to do in this next segment is talk a little bit about the pathogenesis of lupus and how that relates to target selection for drug development strategies. A little review of the immune system, and this is in everybody, not just lupus. There are two types of immune system subsets. There's the innate immune system on the left and the adaptive immune system on the right. And the innate immune system is ancient. It's in fact found in all plant and animal life. The responses are rather nonspecific, so there's immediate defense. It's not the least bit sustained. Examples are the complement system, neutrophils, toll-like receptors. In fact, the toll-like receptor was discovered in a fruit fly. On the right-hand side are characteristics of the adaptive immune system, which is evolutionarily advanced and found in vertebrates only. The responses are antigen specific, and there's memory to mount stronger and stronger attacks via somatic mutations. Examples are lymphocytes and antibodies, but there's clearly crosstalk between the two systems. So with that as background, here's my rather simplistic rendition of the pathogenesis of lupus. These are some of the key components, and we have components of the innate immune system on the top right and the adaptive immune system on the bottom left. We'll take a genetically susceptible host and expose that patient to some kind of trigger. We don't know what the environmental triggers are in lupus. For years, people have been researching viruses. In my cartoon, I have the sun beaming down on the skin of a lupus patient, causing death of skin cells and the release of RNA and DNA. We know that cigarette smoking is a risk factor for some patients. All right, so here's my cartoon. So we have the sun beaming down on the skin of lupus patients. And that the ultraviolet light can cause death of skin cells. Apoptosis is the exact word. And the release of their contents, the RNA and DNA, enters into a plasmacytoid dendritic cell. And plasmacytoid dendritic cells are very abundant in the skin of lupus patients. The DNA and RNA signal the plasmacytoid dendritic cell via toll-like receptors, specifically toll-like receptors 9 and 7. And the result of that is the elaboration of a lot of pro-inflammatory proteins, chief of which are the type 1 interferons. And everything that you see on this slide has been, is, or will be targeted for drug development in lupus. We'll cross over to the adaptive immune system, to the T cell, activation by the myeloid dendritic cell. And then the T cell interacts with a B cell, another lymphocyte. And that interaction is via the T cell receptor and the MHC. But that interaction alone is not enough. There are many what we call co-stimulatory molecules involved. The B cell becomes activated, and to sustain its growth and survival, and in fact its differentiation, it requires certain cytokines. And two are here, APRIL and BLyS. BLyS stands for B lymphocyte stimulator. You'll also hear it referred to as BAFF. In fact, BLyS or BAFF is the target for belimumab. The fate of a B cell is that it can mature into a B memory cell or into a plasma cell. And both B cells and plasma cells make antibodies. And in the case of lupus, autoantibodies. And in this little cartoon, I have an anti-DNA antibody complexing with DNA. And that immune complex can activate the complement system. It in turn attracts neutrophils or polymorphonuclear leukocytes, a type of white blood cell that can injure tissue. And it can also, the complex can also activate macrophages. That little halo is what's called a DNA net. It's released from the neutrophil when it's dying. And this immune complex can further activate the plasmacytoid dendritic cell via a different receptor, the Fc receptor on the PDC. And the DNA net can also activate the plasmacytoid dendritic cell. So this goes round and round and round. All right, in order to understand clinical trial results, you need to know the disease activity instruments. And sometimes explaining these is a bit dry, so I'll try to condense it. I don't use any disease activity instruments when I see my patients. I don't use SLEDAI, I don't use BILAG. I guess what I call it is Gestalt. And if I want to further characterize the patient, I suppose I could use the words mild, moderate, and severe. But I know my patients all very well, and I know who to worry about and who I worry less about. And that's all just through taking all their clinical material and their laboratory results and kind of sticking it up here in my brain and having an idea of how they're doing. But there are formal disease activity instruments. And the two that have risen to the top are the SLEDAI, and that was originated in Toronto. And there's the BILAG, which came from the British Isles. There are composite indices that have incorporated both SLEDAI and BILAG, and those are known as the SRI, the SLE Responder Index, and that came to fruition in a post hoc analysis of the failed phase 2 belimumab trial. And then there's the BICLA, the BILAG-based composite lupus assessment, which was generated during the eprotuzumab studies. Just a brief description of the SLEDAI. It does evaluate 24 lupus manifestations, and there are scores assigned to each of the 24 manifestations. Now, you may argue that some of the scoring is not appropriate. For example, if someone has mild arthritis, that's worth 4 points. But if someone has life-threatening thrombocytopenia, low platelet count, that's only worth 1 point. But be that as it may, this has been around for quite a long time. And one key to using the SLEDAI is that the manifestation has to be attributed to active lupus. For example, rash is worth 2 points. If the rash is inactive, let's say it's discoid lupus that left a scar, but it's no longer active, that would not be scored when you fill out the SLEDAI. We fill out the SLEDAI and then we can tally the scores. And you can see in the middle of this slide a rough idea of what's mild, moderate, and severe. Now, in order to lose points on a SLEDAI, there has to be complete elimination of the disease manifestation. Or if we're talking about a laboratory abnormality, there has to be resolution. For example, if someone has a terrible head-to-toe rash and it gets 95% better with your intervention, well, as long as there's still a little bit of active rash, that has to be scored. And that's one of the problems with SLEDAI. And as far as resolution of laboratory abnormalities, let's say the DNA antibody is very high, maybe on ELISA greater than 1,000, and it goes down significantly, but it's 1 point above the normal. Well, you still have to score it. Now, as far as point reductions and what is clinically meaningful, there's a range. If you ask various investigators, they'll say anywhere from 3 to 7 points. And that was important for the development of the SRI, which I'll show you. All right, and then there's the BILAG. And the BILAG is divided into various domains. And each domain is scored a letter score, A, B, C, D, or E, where A represents severe disease activity and E, they never had activity in that domain. And then within each domain, there are various items, and each item gets scored as not present, improving, same, worse, or new. And that's compared to the last month. Now, there's about 100 individual parameters, so there's no question the BILAG is a bit more cumbersome than the SLEDAI. One of the advantages of the BILAG is that you can see graded responses. So with SLEDAI, the item has to disappear or the manifestation has to disappear. In BILAG, if it improves, then we take that into account. Now, we've made life even more complicated using composite indices. And as I mentioned, the SRI grew out of the belimumab program, and what's called the BICLA grew out of the epertuzumab program many years ago. And then we've had modifications of the SRI as well. The SRI was born in about 2005, and two things happened at that time. Number one is that the results of the phase 2 belimumab study were announced, and it was a failure. And number two, the FDA, recognizing that sponsors were having a lot of trouble developing drugs in lupus, issued draft guidance. And one item in the draft guidance basically said, you cannot declare someone a success or have a successful outcome if one domain improves, but another domain gets worse. Meaning, if someone's skin disease improves, but they ended up having renal disease, well, that's not a success. So the very clever folks at Human Genome Sciences developed the SLE Responder Index based upon the data from the phase 2 failed study of belimumab. And they came out with this, a responder index, a composite index, which was being used in rheumatoid arthritis. Composites were being used in osteoarthritis and Crohn's disease, but they had not been used in lupus. But this kind of fit with the FDA guidance. In order to be a responder, one had to have a 4-point or greater improvement in the SLEDAI score, but there couldn't be worsening. And worsening was judged by BILAG. And in case the BILAG didn't cover all the different manifestations that could worsen, there could be no worsening in the physician global assessment. Actually, there was about 10% wiggle room allowed. In order for someone to be considered a success, all three items that you see here had to happen. Now, a little bit after that, the BICLA was born, and that was based on similar principles where the bilag was the driver of efficacy as opposed to the SLEDAI in the SRI. And as I mentioned, this was born as a result of the program with epertuzumab. And there was reasonable concordance with the SRI, but I'm going to talk a little bit more about some of the discordance that was seen. And that was with ustekinumab or Stelara, and then the TULIP-1 study, which was the anifrolumab phase 3 study. This particular slide was given to me by Matt Cassino, who works at Genentech. And what it shows is the effect size for BICLA on the y-axis and the effect size for SRI on the x-axis. And when I say effect size, merely what that represents is the difference in response rates between the treatment arm and the placebo arm. And what you see is pretty good correlation in some of the studies that were analyzed. Epertuzumab, rontelizumab, anifrolumab phase 3, basically saying that the BICLA and the SRI performed identically. Then along came the phase 2 ustekinumab study, where the SRI worked just great. There was about a 30-point effect size, and we kill for effect sizes that large in lupus. But with the BICLA, the effect size was minuscule. It was about 2 percentage points. So there was discordance, and that's the first time we saw that. And the second time we saw that was with the anifrolumab phase 3 study, the TULIP-1 study. It was in the opposite way, though, where the BICLA worked and the SRI didn't work. So we need to figure out why the discordance, and we have a lot of people working on that. But luckily, TULIP-2, the other phase 3 program with anifrolumab, had nice concordance between BICLA and SRI. Now, there are a lot of other instruments that we use in our clinical trials. We're starting to do more organ-specific assessments. The CLASI is a measure of skin disease. And just like in rheumatoid arthritis, we're looking at joint scores. We measure flares. There's the patient assessment, the patient global assessment. There's quality of life, and I listed just a couple, the SF-36, as well as the FACIT fatigue. And then we also capture organ damage, though in a short trial of 1 year, it's unlikely to see damage accrual. It takes a little longer for damage to develop. Now, we realize that our endpoints are not perfect, and there are groups that are working on other endpoints for studies. This is the Lupus Low Disease Activity, which came out of the Asia Pacific group, and it incorporates some of the items that you know. In order to be considered in a lupus low disease activity state, and what we know about that particular state, if one remains in that state, there's less damage accrual. And damage accrual is the name of the game for any particular disease. In lupus, we want to prevent damage, either from the drugs or from the disease itself. Anyway, so the definition of lupus low disease activity means that the SLEDAI has to be at 4 or less. There can be no new lupus disease activity compared with the previous assessment. The physician global assessment has to be 1 or less, and the prednisone dose less than or equal to 7.5. Now, largely from Europe, a group of investigators got together and called a new definition of remission, the DARIS definition of remission. And when I talk to Ronald Van Vollenhoven, I always forget what DARIS stands for. And then he tells me, remember the fish in, what was it, Finding Nemo? I forget the fish's name, but it's connected somehow to DARIS. So someone will have to refresh my memory about the little hippo tank or whatever it was. Anyway, it's great that we're talking about remission. This was not in the vernacular years ago. And you can see there are many different permutations of the definition. There's clinical remission, a complete remission, clinical remission on treatment, complete remission on treatment. I'm not going to go through all the components, but there's a lot of interest in defining remission, which means that hopefully remission is somewhere near us in time. All right, as far as lupus treatment, what I thought I would do next is talk about some of the news from the American College of Rheumatology. And I'm sure you keep up with all this, and maybe some of you attended virtually. I'm sure you attend EULAR, and there was some news at EULAR, some groundbreaking news, and that's been expanded at the ACR meeting. All right, so we're back to this cartoon, and I'd like to talk about the innate immune system, inhibiting the innate immune system, specifically the interferon pathway, the type 1 interferon pathway. There are 3 types of interferons. There's type 1, type 2, and type 3. Type 1 has actually 5 subtypes: interferon alpha, beta, omega, epsilon, and kappa. And what's key here is that all 5 subtypes bind to the same receptor on a cell, and that's called the interferon alpha receptor. And I don't really like the nomenclature, because then you might think, well, just interferon alpha binds that. But no, all 5 subtypes bind the interferon receptor. We'll call it the type 1 receptor, type 1 interferon receptor. Then there's type 2 interferon, and that consists of interferon gamma, which binds to yet a different receptor, the interferon gamma receptor. And then there's type 3 interferon, known as interferon lambda. And we're actually hearing a lot about interferon lambda because of COVID. Now, the interest in interferons is not new. I mean, look at the date of this New England Journal of Medicine article. It's 1979. And the conclusion by the investigators at the NIH was, it is possible that the production of interferon may contribute to immunologic aberrations in autoimmune diseases. So they were ahead of their time. And that was based upon this survey of patients with various rheumatic diseases. You can see lupus, rheumatoid arthritis, and a few other conditions. And they basically measured interferon in the blood of these patients. What I have highlighted in the red box are patients with active lupus, and you can see at the far right, 71% of the patients had evidence of interferon. So it was known long ago, 1979 is when I graduated medical school, that interferon was playing some role in lupus. Well, we've learned more about interferon in lupus. We know that there are elevated levels of interferon alpha in patients with lupus. If you take the sera of patients with lupus and mix it with normal white blood cells, you can induce an interferon gene signature. And what an interferon gene signature is, it's basically a compendium of all the genes that are turned on when exposed to interferon. It's been very difficult to actually measure interferon in the blood. It's far easier to measure the gene products or the gene activation of all the different genes that are turned on by interferons. Anyway, that's called an interferon gene signature. And we know that about 2/3 to 3/4 of lupus patients have an interferon gene signature. And we also know that clinical and serologic activity correlate with interferon gene expression. But the burning question for the last 20 years has been whether interferon inhibitors can reduce lupus clinical activity. And there are a lot of different ways to target interferons in lupus, and I don't have time to go through all of these. I thought I would focus on targeting the plasmacytoid dendritic cell, the factory for interferons, and then targeting type 1 interferons themselves. I've done a lot of work with what's called BIB059. It's an antibody that targets the protein BDCA2 that you see at about 2 o'clock on this plasmacytoid dendritic cell. This cartoon shows the plasmacytoid dendritic cell, and remember that particular cell type is very abundant in the skin of lupus patients, but it's also found in other organs. It's found in the kidneys, for example, and it's a factory for not only interferons, type 1 interferons, but also type 3 interferon, as well as other pro-inflammatory proteins, pro-inflammatory cytokines, such as interleukin-6 and TNF-alpha, as well as a variety of chemokines. The protein on the surface, BDCA2, is very interesting. When the antibody to BDCA2, and that antibody again is known as BIB059, binds to BDCA2, the protein on the surface is internalized. And once it is internalized, the PDC shuts off synthesis of all of these pro-inflammatory proteins that you see on this slide. The other thing that this antibody does, the tail end of the antibody binds to CD32A, and that's depicted here at about 10 o'clock on the PDC. And that's an FC receptor. The FC receptor is responsible for bringing in immune complexes and yet another way to activate the plasmacytoid dendritic cell. So in short, this particular antibody, BIB059, has a dual mechanism of action. It causes BDCA2 to be internalized, it blocks the FC receptor, and that results in a downregulation of all these pro-inflammatory proteins. So we did a phase 1 study, a small study in about 12 patients with lupus. 4 of them got placebo and 8 got the drug. And I must say, there was more biology than there were patients, but the biology set the foundation for what I'm showing you now, which was the phase 2 program. And there were 2 phase 2 studies. LILAC-B, which was a study in skin lupus, cutaneous lupus, and Vicki Wirth presented these data not only at this year's ACR meeting, but you saw a preview of the data at EULAR in June. And in order to get into the study, one had to have active cutaneous lupus. It was dose ranging to try to find the best dose that would treat the condition. And if you look at the bottom bullet, there you see the results using the CLASSI instrument that I mentioned before. The placebo patients got better, but only to about 15%. Whereas those treated with the drug, BIB059, about somewhere around 40% on average across the 3 groups, improved. And this is very statistically significant. And it's not surprising because there are a lot of plasmacytoid dendritic cells in the skin. And if this is a way to inhibit the production of their products, then you might expect the skin disease to get better. And in fact, that's what was seen. Now, at the American College of Rheumatology meeting just a couple of weeks ago, I presented the companion study called LILAC-A. And this was the use of BIB059, but in global lupus. In fact, in order to get into this particular study, one had to have arthritis and rash. And we just compared placebo to 450 milligrams of BIB059. And when I say placebo in all these studies, it's true, it's placebo matched to the experimental drug, but the patients come into the studies on their background therapies. We do not make them, for the most part, stop their steroids or stop their immunosuppressives. They just come in on whatever they've been treated with. The primary endpoint was a reduction in the arthritis, in the tender and swollen joint count. And we saw a statistical reduction. We also looked at that global measure of response, the SRI, and there was a very significant difference between the treatment arm and the placebo arm. And we also looked at the CLASSI again, and we didn't see as profound a response as shown in the other study, but these patients didn't have as much rash as in the other study. So with 2 positive phase 2 studies, no doubt this program will advance to phase 3. All right, now let's talk about a different drug, and that's anifrolumab. Anifrolumab is an antibody that binds to the type 1 interferon receptor. So it interferes with signaling by all 5 subtypes of the type 1 interferons. And we saw great data, wow, it's probably about 4 years ago now, phase 2 data with anifrolumab in extrarenal lupus. It worked tremendously well in skin disease. In fact, everything worked so nicely in that particular study. There was a lot of confidence going into the phase 3 program. And the phase 3 program had 2 studies. One was called TULIP 1 and one was called TULIP 2. And you can see that in TULIP 1, there were 2 doses of anifrolumab compared to placebo on top of standard of care. This is given intravenously every 4 weeks. The study was 1 year in length. And there was TULIP 2, which was pretty much very similar, except the lower of the 2 doses was eliminated. Now, the TULIP 1 data were announced, and to the shock, I must say it was incredibly shocking because we were so confident with the phase 2 data. But the short of it is that the TULIP 1 study failed to reach the primary endpoint, which was that composite index known as the SRI. But it did work with the different endpoint, the BICLA. And I mentioned that before when I was talking about concordance and discordance between BICLA and SRI. But we still don't understand why the discordance, but the fact of the matter was that the BICLA worked. So it got everybody thinking, well, TULIP 2 is still underway. So TULIP 2 was a little behind TULIP 1. The data had not been unblinded. Should we be cautious and change the endpoint to the BICLA from the SRI? And the consensus was, yes, let's do that. Well, what you see on this slide are the effect sizes for the 3 studies. MUSE is the phase 2 anifrolumab study, and then the 2 phase 3 studies. And I have the 2 composite endpoints, the SRI and the BICLA. In MUSE, the SRI was the primary endpoint, and you see a nice robust effect size of 22%, and the BICLA worked just fine. In fact, it worked better. In TULIP 1, however, a 4% effect size was not statistically significant. Yet the BICLA at 16% was or would have been statistically significant. And then in TULIP 2, we changed the endpoint to the BICLA, but it wasn't necessary because the SRI worked just fine in TULIP 2. So one of our challenges is to figure out why the discordance. So where does anifrolumab sit? It sits with the regulatory authorities. Hopefully sometime next year, we'll hear whether the FDA approves this particular drug or not. My own feeling, if you look at the totality of the data, not just these composite endpoints, but if you look at the effects on joints, the effect on skin in particular, the drug is quite effective. Now at this year's ACR meeting, it wasn't as sensational as last year. At last year's ACR meeting, I presented the results of the TULIP 1 study and Eric Moran presented the results of the TULIP 2 study. At this year, what was shown, and there were about 6 presentations, was to combine the data from TULIP 1 and TULIP 2. And because they were pretty much identically designed, one can combine the data to generate more statistical power. And these are some of the questions that were looked at. Well, one is actually the phase 2 study, what happens to patients when they stop interferon inhibitors at the end of the study, and the patients got worse. That's the short answer. I presented, what does it mean to be a BICLA responder? These endpoints are very complicated. I mean, it's complicated enough to deal with SLEDAI and BILAG, and then to incorporate them into these composites. So I had a poster on what it meant to be a BICLA responder. We looked at flare reduction and corticosteroid tapering, and the short of that analysis was that anifrolumab was not only associated with a reduction in flares compared to standard of care, one could taper the steroids more effectively on anifrolumab. And if you look at both endpoints, flare reduction and the ability to steroid taper, anifrolumab was superior. Herpes zoster shingles was examined because we do see a higher frequency of shingles in patients who get anifrolumab. It's about 5 to 6% versus 2% in the standard of care arm. And this was an analysis of the patients who developed shingles. We looked at the efficacy across various organ systems. It's important for a drug, or ideal, I should say, for a drug, to be able to treat all different manifestations of lupus, to treat all different types of patients, and that's what this analysis showed. And then there was a poster about the early and sustained reduction in the severity of the skin disease. Anifrolumab works very quickly on the skin, like within a month or two. I mean, the endpoint for that particular study was for the studies was week 12, but it did work very quickly. All right, now we're going to switch gears and talk about targeting B cells. So remember that B cells are lymphocytes that do a couple of different things. They make antibodies, and in the case of lupus, autoantibodies, but they also make a variety of pro-inflammatory mediators, cytokines, and they also interact with other cell types. So you can see that in lupus, there are a lot of potential targets. I don't have time to study them all. CD22 was eprotuzumab, but that program ended a while ago. But there's still a lot of interest in antibodies to CD20, and CD20 is on B cells, and an example of a CD20 antibody would be rituximab. Rituximab kills the B cells. There's also interest in some of these, what we call nuclear transcription factors, also known as Ikaros and Ilos. And then there has been interest in Bruton's tyrosine kinase and inhibitors of that particular protein, because it is crucial for B cell function. All right, obinutuzumab is a third generation, well, you can think of it as a third generation rituximab. It's far more potent at killing B cells. Why is that important? Well, we think the failure of rituximab long ago in the lupus nephritis, as well as the extrarenal trials, was because there was inadequate B cell depletion by rituximab. Obinutuzumab has been approved in the United States for the treatment of chronic lymphocytic leukemia since 2013. It is a far more potent B cell depleter than either rituximab or ocrelizumab, or for that matter, ofatumumab. So the feeling was, well, maybe we'll do better in lupus if we can deplete the B cells more effectively. And I showed some of the lupus nephritis data a year ago at ACR, and at this year's ACR meeting, I showed the 2-year data with obinutuzumab. And what you see here, so we're switching gears, we're talking about lupus nephritis, and these are renal response endpoints, what we call the complete renal response, which is what we want to achieve in lupus nephritis. And you see 3 time points, 1 year, 1.5 years, and 2 years. And basically, the slide shows a comparison between obinutuzumab versus placebo on top of mycophenolate. Everybody got standard of care with mycophenolate and steroids. And you see a nice effect size of 12% at 1 year, which actually grew to 20 percentage points at 1.5 years. And for lupus nephritis, I mean, if you ask rheumatologists and nephrologists, they'll tell you they'll settle, I mean, barring any safety issues, for a 10% effect size. We just need to do better in lupus nephritis, no question about that. Because right now we're just achieving complete response in not even a third of our patients. And we know that the patients who do best long-term are those who do best short-term. So at 1.5 years, the effect size grew to 20%. And at 2 years, it was about the same. But what's so striking, I didn't show you the schedule of treatments, but the treatment was 2 infusions of obinutuzumab 2 weeks apart at the very beginning at week 0, and then another course of therapy at week 24 at 6 months. So what we're seeing is an effect that lasts 1.5 years after the last exposure to obinutuzumab. So that's what I'm very impressive results. And there were no safety issues that were seen. All right, I'm going to switch to a different drug and a different strategy, and that's iberdomide. Iberdomide has another name, it's called CC220. And this affects those transcription factors that I just showed you within the nucleus of the B cell. Basically, it accelerates their degradation. And those particular transcription factors are very abundant in inflammatory cells. So this is a strategy for reducing inflammatory cell numbers and activity. And what's so striking about lupus drug development, and I use the word eclectic, is that lupus is being attacked from so many different angles with so many different mechanisms of action. All right, so what you see here, and this is presented by Joan Merrill, the results at 6 months using the SRI-4 as the response endpoint. Placebo was compared to 3 doses. And if you look at that top line called SRI-4 primary analysis, focus on the numbers in the parentheses. Those are the percentage of patients responding. And you see about 35% in the placebo arm, and at the highest dose, 54% in the iberdomide arm. So that's an effect size of 20%. And as I mentioned, in extrarenal lupus, I think anything over about 10 to 15% is exciting. But it gets even better. We'd love to move towards an ability to identify patients who would be more apt to respond to a drug intervention. So what we call personalized medicine. And we've really not been able to do that in any of the rheumatic diseases. I mean, you would think in rheumatoid arthritis, by this time we'd be able to somehow know who should get which rheumatoid arthritis drug, or in psoriatic arthritis or psoriasis, who would respond better to one drug over another. But we're not at that stage. But this looks at that issue. And so if you look across the very bottom of that graph, you'll see overall Aiolos low, Aiolos high, interferon low, interferon high. So those are the markers of the patients who had elevated levels of the particular targets, Aiolos, or there was another target, Icaros. But focus on Aiolos. And you see greater effect sizes. If you look at the histograms number 2 and number 3, in particular Aiolos high, histogram number 3, there's a 33% effect size. So the effect size grows. The overall is on the far left, the Aiolos high is in the middle, and you can see that the effect size grows, so that maybe this particular therapy should be used in patients who test to be Aiolos high. Now, it also seemed to have benefit in those who were interferon high as well. Anyway, so more work will be done as this program moves from phase 2 to phase 3. Well, there was disappointment with BTK inhibitors. I showed you BTK on that cartoon. It was a novel way to inhibit B cell function and B cell numbers. And here you see evobrutinib results. And last year, David Eisenberg presented BTK inhibitor called fenebrutinib. The study from last year, the study from this year, both failed. When a study fails in lupus, you never know whether it's the drug or the clinical trial design. But I think in this case, we have two of very similar drugs failing. So it's probably not the right strategy. All right, let's move to extracellular targets. We'll focus on the right-hand side of this slide. As I mentioned early on, Bliss and APRIL are key growth factors for B cells. And we think that eliminating B cells is good for lupus. Bliss and APRIL function through Bliss receptor, TACI, and BCMA on the B cell surface. And there are different ways to inhibit Bliss, but the focus is on an antibody to Bliss. And you know that antibody as belimumab. But another program that you might follow is the TACI program. TACI being the receptor, or one of the receptors, for Bliss and APRIL. And there are development programs with TACI for extrarenal lupus. But I want to talk about belimumab. At EULAR, I presented the Bliss LN. Now, we go back a lot of years ago when we started the Bliss program, or I should say the belimumab program. It was actually probably about the year 2000 with a phase 1 study, and that served as the foundation for phase 2. The phase 2 study failed, but there was perseverance, and we learned from phase 2 to inform phase 3. The 2 phase 3 studies I showed you, they were both successful, and that led to yet other studies, most of which were successful. A subcutaneous study with belimumab, the Northeast Asia study with belimumab, both successful. There was a study of African Americans that failed, but the effect size was just a little bit less than what we typically saw with the other studies. But what we saw in a post hoc analysis from the belimumab phase 3 was that there seemed to be benefit to patients who had low-grade renal disease. Years ago, we started the Bliss lupus nephritis, Bliss LN study. And again, I don't have enough time to go through all the details, but it had a lot of unique features. Belimumab versus placebo was given to patients with either proliferative nephritis or membranous nephritis or both. And they could receive as background therapy mycophenolate or cyclophosphamide. That was a very unique feature. And that was up to the investigator, whichever drug they were comfortable with. And there were some other unique features as well. I'm showing you the top line data, and the primary endpoint was measured at 2 years, which was also very unique. Usually lupus nephritis trials go for 1 year. And the primary endpoint was known as the primary endpoint of renal response, PER for short. In yellow are the response rates, or orange down the center, and then the effect size in the column just next to it. And on the far right, the p-value. So the primary endpoint was statistically significant, and all the secondary endpoints were statistically significant. So that was presented at EULAR. What was presented by me at ACR was a further dive into the data. I mean, there's a huge amount of data that's been accumulated for Bliss LN. So the data will come out over probably the course of a couple of years. So we looked at the difference between background therapy, whether patients got mycophenolate, that should be MMF, not MMV, versus cyclophosphamide. We reported on the response rates in the various histologic classes, proliferative versus proliferative and membranous versus membranous. We looked at race, we looked at the steroid sparing, and we looked at biomarkers. Now, the hope is that the FDA will rule on the extension of the belimumab label to lupus nephritis shortly. I mean, I hope within the next month or two. And these are some of the data that I showed. I don't know that we have time to go into detail, but on the far left, we have the treatment regimen, what kind of background therapy they received, the lupus nephritis class, and race. And if you look to the right, that's what's called a forest plot. And you have the point estimates to the right of that vertical dotted line means that there was numerical superiority of belimumab over the standard of care. And all the points are to the right except for the class 5, the pure membranous. And pure membranous is always an issue in these studies. Not that too many lupus nephritis studies have allowed pure membranous in, but there are also some data, which I don't have time to show, suggesting that belimumab may actually prevent worsening. We may prevent flares in that pure class 5 group. And if you look at the far right, you have the p-values for these various subgroups. The problem in looking at subgroups in any study is that they are underpowered. When you start carving up the data and the groups start getting smaller and smaller, one loses statistical power. So one has to be a bit careful in evaluating subgroups. All right, let's move on to a different strategy, and that is inhibiting the plasma cell. So remember that the B cell can differentiate into a B memory cell or into a plasma cell. And the plasma cell makes antibodies, and in the case of lupus, autoantibodies. We've not targeted to any major degree the plasma cell with any of our therapies. Even rituximab or obinutuzumab those therapeutic antibodies don't reduce plasma cell numbers. Why? Because the target for rituximab and obinutuzumab is CD20, and CD20 is shed from the surfaces of plasma cells. But there are other ways to inhibit the plasma cell, and Reinhard Wolf's group reported the effects of bortezomib many years ago. Then there was a trial with ixazomib, except that trial in lupus nephritis was prematurely terminated because of a failure to adequately enroll patients. But underway are studies with KZR616, which is a proteasome inhibitor, an immunoproteasome inhibitor to be specific. And this is a way of inhibiting the plasma cell. So at the ACR meeting, I presented a poster which basically had the first part of the study, about 36 patients with extrarenal disease in an open label design, showing the effects of this particular drug. And then there were 2 patients with lupus nephritis. So we're now transitioning to lupus nephritis, and we'll see how those data go. Targeting T cells, or T-B cell interactions. This interaction between these 2 types of white blood cells is very crucial. A lot of targets on T cells and a lot of targets on B cells to prevent that interaction. But I'm going to tell you about voclosporin. You've heard probably of cyclosporin. Well, voclosporin is just 1 amino acid different. And it's far more potent than cyclosporin. And it is a little easier to control because of the way it's metabolized. Well, voclosporin was tested in a phase 2 study, and it showed efficacy in lupus nephritis. There were some potential safety issues which were actually put to rest in this phase 3 study. The phase 3 study in lupus nephritis is known as Aurora, and data were first presented at EULAR. It was a 1-year study with a 1-year endpoint, and the primary endpoint was renal response. And in both the belimumab as well as the voclosporin studies, in fact, in all lupus nephritis trials, the primary endpoint is driven by a reduction in proteinuria. And here you see an effect size of about 18 percentage points. Voclosporin patients, about 41% responded to that level, and 22.5% in the placebo arm. Again, not just placebo, but background consisted of steroids and mycophenolate. And then you can see all the secondary endpoints hit. So my prediction is that we'll see approval of voclosporin sometime in early 2021 for the treatment of lupus nephritis. Now, there was nothing very exciting at the American College of Rheumatology because all the excitement existed at EULAR. But Ellen Ginsler had a poster where the phase 2 and the phase 3 studies were combined. And if you look at the bottom of the poster, you can see the 6-month data on the left, and on the right, the 1-year data. And there's a very impressive effect size of 20 percentage points. What that means is about just 1 in 5 patients will benefit from being on voclosporin. Now you may say, well, 1 in 5 is not very much, but if you can prevent worsening of kidney disease and have a better outcome in an extra 20% of the patients, I think we've made major headway. All right, and just to finish up, I was asked to mention why all the failed trials. Well, actually, we're reversing that. So you'll notice we had a whole bunch of positive trials announced at the ACR meeting. We had belimumab in lupus nephritis. We had voclosporin in lupus nephritis. We have BIB059 in cutaneous lupus and regular lupus. And what else am I missing? Oh, obinutuzumab in lupus nephritis. We did have some failures, but it used to be in the olden days, everything we studied failed except for belimumab. So some thoughts about why all the failed trials? Well, you never know if a trial fails, whether it's the drug, the design, or it's the designers. I think one problem that we face is that lupus activity is overstated. In order to get into the extrarenal trials, one has to have a certain amount of lupus activity, and it usually comes down to a certain number of SLEDAI points, so like 6 points or 8 points, or maybe a certain amount of BILAG activity. The investigators want their patients in these studies. The patients want to get into the studies. So there's a tendency, I think, to overstate lupus activity. For example, rash. Rash could just be a little redness of the cheeks, and that's worth 2 points on the SLEDAI. Arthritis. If you have 2 joints that are painful or swollen, that's arthritis worth 4 points. There are 6 points. If you have hair loss, alopecia, that's 2 points. I'd say 99% of the women that I ask, do they have hair loss, say, yes, they have hair loss. When they shower, there's a little bunch of hair down in the drain. Well, that's not really what we meant in scoring alopecia on SLEDAI. And then oral ulcers is a major source of contamination. We're not talking about canker sores. We're talking about palatal ulcers in the lupus patient representing disease activity. So some of these SLEDAI items are incredibly subjective. And if someone enters with really minimal activity, but has the points, they're going to get better with the placebo. So you really need lupus activity. And in fact, there have been analyses. The first time we did this actually was published by Ronald van Vollenhoven many years ago. It was a post hoc analysis of the phase 3 belimumab program, where they looked at patients with high disease activity. That is, patients with low complements and high DNA antibodies, which essentially was a surrogate for high clinical activity. And that represented about 50% of the patients in this study. And those 50% had a much greater effect size. The effect size grew from about 11 percentage points up to 20 percentage points when the focus was on those with high disease activity. All right, another reason that we might have failures is the background therapy that's used. We allow patients to come in on background steroids, on background immunosuppressives. But that blunts our ability to discriminate between placebo and drug. And that was shown many years ago in the Explorer trial That was the study, an extrarenal trial of rituximab. Patients came in on immunosuppressives. Their steroids were boosted up from whatever they were on to either 45 or 60 milligrams based upon their entry dose. If you treat someone with enough background therapy, you're not going to be able to prove that your experimental therapy is a success. We also know that there are multiple pathways that are active. I think far more than in rheumatoid arthritis or in psoriatic arthritis. And maybe we need to target not just one particular molecule, but many molecules. Though I must say, when you have someone coming into the study on hydroxychloroquine, steroids, and an immunosuppressive, we already are targeting multiple molecules. But you'll hear more about multiple targeted therapies. All right, the endpoints may not be optimal. Well, as I mentioned, SLEDAI and BILAG have risen to the top, and then we've incorporated them into composite indices, the SRI and the BICLA. And while they may not be optimal, they are what we have and what the regulators accept. There certainly are groups, as I mentioned before, working on other endpoints that could be useful in clinical trials. There are other key questions to answer. Is the biology the same for all manifestations? That is, should we be doing organ-specific studies? Does the skin have the same biology as the kidney, and the kidney the same biology as the joints? Are pathways more restricted for certain manifestations? Well, we've seen marvelous skin responses with anifrolumab. So we know that the interferon pathway is activated in patients with skin disease and lupus. Do we need to target more targets? So our bispecific molecules, and a bispecific molecule, let's say a bispecific antibody, the two heads of the antibody, one binds one molecule and one binds yet a different molecule. So might that be the answer? And there are studies underway. There's one from Amgen with an antibody to ICOS ligand on one side, and the other half of the antibody inhibits BAFF or BLyS. There are studies with dual BTK and JAK inhibitors. And then you might hear fairly soon about a sequential rituximab and belimumab study. How can we improve upon trial design? Well, I could go on for hours about that. How can we eliminate a lot of the noise? How can we enroll patients with truly active disease that are informative? Do we need biomarkers to help guide us, as I showed you with iberdomide? And one area where we've really failed is the patient-reported outcomes. We have not seen a good correlation between patient-reported outcomes and our clinical endpoints. All right, but we are awaiting news. And if I didn't list your favorite study, I apologize. So news from sponsors. We should hear, I would say within a year, about the baricitinib phase 3 study. That speaks to JAK inhibition. And then Boehringer Ingelheim did a study with an antibody to CD40 in lupus nephritis. And I would say in the next 3 to 6 months, we should hear about that. And there's many other studies that are underway in earlier development. In fact, I mean, it's unprecedented, the amount of activity in lupus these days. And then news from the FDA. We should hear about both belimumab and voclosporin within the next month to 2 to 3 months, somewhere in there. Both phase 3 studies being successful. So I am optimistic. I think we will have 2 new drugs next year for lupus nephritis. And then actually I should also put on this the anifrolumab FDA decision. That will come later in the year, maybe around mid-2021. And I'm optimistic about that as well. So I am optimistic about the future. I think we will have many more medicines. I think someday, and maybe that day is sooner than later, we will have biomarkers so that we can individualize therapy. And all this will lead to better outcomes for our patients. I mean, if you go back to pre-steroid era, so compound E, cortisone, was developed in 1948. And prior to that, the outcomes for our patients with rheumatoid arthritis, for lupus, were miserable, especially for lupus. There's about a 50% 7-year survival. But you see the survival improve each era. So with the steroid era, then the immunosuppressive era, and the biologic era. But we still have a ways to go. We want to see 100% survival, and we want to eliminate all comorbidities. And I think the time will come. We are making progress. It's been slow, it's been frustrating, but we've persevered. It's been incredibly humbling. I've been involved in this area for about 25 years, but we are starting to see the light at the end of the tunnel. So I thank you very much. And now we'll take on some questions.